for this, and then can everybody hear me? I don't think you can hear anyone.
Yes, I can hear you.
We can hear you.
Awesome. All right, so everybody hears me. Hello, this is how I chose to defend my PhD incarnated in a virtual world. Because, well, in this way, you know, it feels more like I'm presenting in front of an audience in an amphitheater, and I think that you can see my body language, which is also a very important part of presenting any work. All right, so thank you very much. So I'm here today to talk about the work I have done during my PhD, which is computational approaches to developmental biology. And so I'm gonna explain a little bit why I titled my PhD that way. But today I'm gonna focus a bit more onto just one of the different stories that I've done, which is the origin and evolution of Oskar. All right, so before I start, I just wanna try to talk a bit about biological system. Because biological system are complex system. If we take an example like a gene, right, which is the basic functional unit of a living system, this gene will translate into a protein which can go and regulate other proteins and other genes. And this protein can also interact with other proteins, creating even more complexity. But this complexity is not restricted to the inside of a cell, because cells will communicate information to coordinate actions, such as creating an organ or communicating between different cells. And this extends even all the way to the individuals, which are interacting within a complex environment. And those species will influence each other's, creating a complex map of interaction in their ecosystem. And so complexity is really seen at all levels of biology. And to try and decipher this complex, those complex systems is really what motivated my research. And I'm just a human, so I can't really study all of biology's complexity. So I had to focus things, and in Cassandra's lab, the focus is really on arthropod diversity and developmental biology, right? And so arthropods are a very diverse group of animals, which comprise things like the Chelicerata with arachnids and scorpios, but also the Myriapoda with the myriapods, crustaceans like crab and lobsters and shrimps. And then we enter the insects with the three main groups of insects, the Ametabola with silverfish, Hemimetabola with crickets, cockroaches, praying mantis, and other animals. And then Holometabola with the well-known Drosophila fly, but ants and wasps and butterflies as well, right? So the diversity of morphology and developmental mechanisms in arthropods is so vast that it makes them an excellent system to try to understand how those developmental traits evolved, right? But to understand that evolution, we need to be able to measure those traits and a lot of other parameters in multiple organisms. And this is sort of where the time of I started my PhD matters, because the last decades has seen revolution in the way that we measure data about biological systems. The genomic revolution led to the sequencing of hundreds of arthropod genomes, and the subsequent transcriptomic revolution to the sequencing of thousands of transcriptomes of arthropods, right? And so this now allows us to understand the evolution of genes, genomes, and organisms with a much higher statistical power and level of understanding. Moreover, the accumulated knowledge of researchers over the last centuries is now sorted, classified, and made available in large databases project, right? And using those databases, we can then link previously disparate biological data and apply this knowledge to large-scale analysis. Not only can we now study the evolution of genes, but we also now can study the interaction between those genes at a sort of global scale, with the creation of screening technologies that allow to map the interaction between all proteins in multiple organisms, such as yeast or humans, right? And so using those interactions, we're now able to better understand the intricacy of the regulations happening within those cells. And similarly, we can read the transcriptomes of single cells, right, which allow us to better understand how the regulation happens internally, but also between cells at a single cell resolution, right? And this applies as well to microscopy, where advances in microscopy now allow us to record embryos developing and recording all the cells in an embryo, right? Which now allows us to perform analyses that were not previously available, such as comparing with morphometric the shapes and the variance and shapes of positions of cells between embryos of the same species or between embryos of different species. All right, and so all of those approaches, they have one thing in common, which is that they generate enormous amount of data that can only be analyzed with the help of computers. And my PhD was really centered around the use of different computational approaches to try to answer developmental biology questions in arthropods. And so the first project that I proposed was a very ambitious one, Cassandra, you're right. It was to use single-cell RNA sequencing and light sheet microscopy to study the germ layer specification in the early embryogenesis of Parhyale hawaiensis, which is a crustacean. Then I worked on a collaboration with a postdoc in the lab called Tarun Kumar. And together we studied the regulation of the organogenesis of ovaries in Drosophila. And by using the network of protein interactions that I just talked about, we studied the regulation of the number of ovarioles, right? And the number of eggs laid by flies. And this work is published in eLife if you want to know more about it. Then I took a little detour in virtual reality, where I am right now, to create an open source tool to visualize 3D plus time light sheet datasets. And then finally, I studied the origin and evolution of the gene oskar. And this last story is really what I will present today. And the first part has been published in eLife, so you can read more about it there. And the second part is currently in its near final manuscript format and will soon be sent to journals. I want to emphasize that the second part is the result of the mentoring of Savandara Bess, which was a highly talented master student who worked with me for 6 months. Okay, so let's talk more about the origin and evolution of oskar. But before I tell you about oskar, I need to introduce a particular lineage of cell called the germline. So the main way that life reproduces is through the replication of its genome, right? A cell will divide in two, leading to two daughter cells. And this process will just repeat as long as resources are available. Therefore, for a unicellular organism to pass down their genome to the next generation, they only have to divide themselves. But when single cell aggregated into multicellular organism, a new problematic emerged, which is that which one and how will that multicellular organism separate itself to reproduce and to continue to propagate the organism's genome. Right? And so during the evolution of multicellularity, there is a solution that emerged which required the separation of fate. So some cell here in blue, right, will live and divide within the organism, and those are called the soma or somatic cells, and other cells here in red will separate, right, and form a unique cell, and from this unique cell, they will recreate a complete multicellular organism, right? And this lineage is called the germline. And so the germline is, and so to know, and so one of the question that emerge is, which one of those cells from the multicellular organism will gain the privilege to actually become the germline, right? Which one will gain the privilege to be the one to pass down the genome to the next generation? Because for all the somatic cells, to not reproduce means that their evolutionary trajectory ends with the death of the organism, okay? And so, so in order to specify the germline, organisms have evolved two main specification strategies. And those are commonly named inheritance and induction, right? And so the inheritance specification strategy will start inside the egg, where germline specification material here in red, like those granules here, right, will be deposited in the egg and is often composed of RNA and protein. And often during the first division of the embryos, there will be an asymmetric inheritance of this material to only a small population of cells. And later during the development, the cells that have engulfed that material will then change their fate and differentiate into germ cells. The second mechanism is the induction mechanism, which uses a slightly different strategy because the egg does not contain any specification material. And during the early development, there is no detectable other type of material that is expressed. But at some point during the development, right, a population of cells will start to send a signal to a smaller population of cell. And that population of cell that receives that signal will then turn and change its fate onto a germline cell. And in both cases, we end up with a separation of fate of soma and germline, and then the cycle will just continue. All right. And so for the rest of this talk, I'll just be using those two icons to differentiate inheritance and induction. Okay. All right. So insects are a particularly great system to study the evolution of those strategies, as both strategies can be found within different lineages. But before I tell you about the strategy, let me just introduce the three main groups of insects. So Ametabola, which I talked about before a little, are insects where the development happens with no metamorphosis. What that means is that the egg hatches in a animal that has its adult form. It will not change further from there. Hemimetabola insects are insects where the development happens in two stages. First, in the egg, then it will hatch as a nymph. And that nymph often resembles the adult, but then through molding, right, this nymph will metamorphose slightly into its adult form. And finally, holometabola, which has where the development happens in three stages, where the egg will hatch as a larva, and then this larva will enter a pupal stage where the organism will undergo drastic changes to metamorphose into its adult form, which often bears no resemblance to its larval stage. And by this, a good example is, for example, is butterflies, where the caterpillar, right, resembles nothing as the butterfly that emerges after the chrysalis form. All right. And so to go back to germ cell specification, in insects, right, hemimetabolous insects like cricket will have a germ cell specification strategy, which is an induction strategy, okay? But in holometabolous, their insects, there is a derived form of germ cell specification which emerged, which is an inheritance mechanism, right? And in multiple instances in holometabola, this derived inheritance mechanism has reverted back to the induction mechanism. And so by studying what mechanism sort of led to the evolution of the inheritance mechanism here, but also maybe like the reversal onto the induction mechanism in other animals, insects provide us with a really good evolutionary model system to study how those germline specification strategy evolve. So I'm gonna talk a bit more about the inheritance mechanism because this is the one that is best known in insect and also the one that is derived. So it's kind of the one that we were gonna try to find a signal for. And I'm gonna focus now on Drosophila melanogaster because this is where this mechanism is best understood. And so in melanogaster, the germplasm is deposited at the posterior pole. And so the germplasm is what I have called the germline specification material before. So in insects, we call it germplasm. And then at the posterior pole where the germplasm was, you will have the formation of a population of cells that will separate from the rest of the egg called the pole cells. And those cells will then go on to become the germline of Drosophila. Okay. And so in order to find that this material called germplasm here is actually what induces germ cell in Drosophila, the first experiment that was done was to take aspirate the germplasm from one egg and then inject it into another egg, and then to wait and see what happens. And when this experiment was done, what they saw was that in the anterior pole where they injected the germplasm, pole cells will develop, meaning that the germplasm seems to be the germline determinant and at least sufficient to induce germ cell determination. And so in order to further study this, researchers tried to find which gene might be regulating this germplasm. And this is where oskar comes in. because oskar was found to be localized at the posterior pole of the embryo, but it also was found to localize in the germplasm. And finally, it was found to be engulfed within the pole cells. So this gene was a good candidate for a genes involved in this germ cell specification strategy. And by inducing an over oskar expression, right, the amount of germplasm actually increased, and this led to an increase in the amount of pole cells that would form in the embryo, okay? So this meant that oskar in some way was controlling the nucleation of this germplasm material here, which then in turn led to more pole cells being produced. And when researchers mutated oskar and made an oskar mutant that was deficient in oskar, they found that there was no germplasm, and they found that there was no pole cells. So it seemed that oskar was the one gene that was at least necessary for germplasm formation and germ cell determination. When they did another experiment, which is that they mislocalized oskar at the anterior pole of the embryo, they found that germplasm would form at the anterior pole along with pole cells later on. So this meant that oskar was sufficient for germplasm formation and germ cell determination. So, so what about oskar in other insects and its relationship to this germline inheritance mechanism that we talked about? So there's very few studies that have studied the effect of oskar in other organisms than Drosophila, but it seemed that in holometabola at least, the absence of oskar correlates to a reversal onto the induction mechanism. With the strange states of Lepidoptera, which it's still unclear whether they use germplasm or an induction mechanism or both. But this hypothesis that oskar seems to correlate with the inheritance mechanism is based on very few studies, right? And so this is sort of where I started my PhD, with very few known oskar sequences and a crucial role that it seemed to play in the evolution of a very important mechanism in holometabolous insects. So I decided to go and search for oskar ortholog, right? And this is where I worked based on the work that was previously done by Tamsin Jones. So Tamsin was a grad student in the lab when I started, and she had manually curated a collection of about 40 oskar ortholog. Then I built a custom pipeline to try and find more sequences based on that first collection. And this is where Savi Savandarabes arrived in the lab, and then she expanded this pipeline even further and worked on automatizing more steps. And so for the rest of this talk, whenever I'm speaking about something that was done either in collaboration or relying on the results with one of those two persons, I'm gonna put a little icon photo at the bottom here. Okay. So we started with oskar DNA sequences, and then we translated them into amino acid sequences. And the reason we did that is that amino acid sequences are much more stable in evolutionary time, which makes the search for orthologous sequences much easier because of the deviation that random mutation will create. So we then align those amino acid sequences into a sequence alignment, and then we took the sequence alignment to create a HMM model of that particular alignment. And those HMM profiles are really good tools to define a particular gene. Because they will basically create a probabilistic representation of the amino acids at different positions in the gene and their transition probability between different positions and within their own position. And so this is a much more powerful way to represent what a gene is than say BLAST, for example. Okay, so we took those HMM profile and then we went onto NCBI and we downloaded every arthropod genome and every arthropod transcriptome, which were like thousands of them. And then we just ran a search using those HMM profile for the gene oskar. And then we got our oskar hits that were like potential candidates. Then we took those and we converted that to amino acid, and then we made a sequence alignment, and then we regenerated an HMM profile and repeated the cycle, right, until we could find no more oskar sequences in the genome and transcriptomes that were published. Okay, and so this led to the creation of a data set of about 369 oskar ortholog. And for the rest of this talk, I'm only going to focus on the protein sequence and the sequence alignment because we didn't do anything with the DNA sequences. Okay, so the first question, right, that we wanted to answer was like, when did oskar appear? Because as we have seen before, oskar was found as early as hemimetabola, but given the low sample size of tested genome and transcriptome, we could here use a large collection of sequences and try to answer, this particular question. And so when we actually look at where do we find oskar in the tree of evolution of arthropods, right, it seems that it is restricted to insects. So the previous hypothesis that oskar is restricted to insects seems to hold true. However, we find it earlier than hemimetabola because in some ametabola insects, we find oskar. So if we zoom in a little, we actually find that oskar seems to have originated in the branch leading to the divergence of zygentoma, which are silverfish insects. All right. So the only thing is that I can't really rule out that oskar is not present in Collembola or Archaeognatha just because of the very low number of available data sets. So it's hard to conclude that oskar is not present there. It might just be absent because there is not enough data, or and as more transcriptomes are published, we might actually find an oskar sequence. However, we're very confident that oskar is not present in non-insect span crustacea because we have tested thousands of transcriptomes and genome there and found no sign of oskar there. So it seems at least that oskar is restricted within this area. Okay, so that means that oskar is a new gene, right? Because the definition of a new gene is a gene that has no ortholose sequences outside of a defined phylogenetic clade. Okay, so how do new gene appear, right? And there exists multiple mechanisms, but the four main ones are gene duplication, where a particular sequence can get copied in the genome, carrying over its gene, right? And then that new copy of the gene is then free to mutate, and through random mutation will then acquire potentially a new function, and then go on and act as a new gene. The second mechanism is rearrangement, where only part of a gene will actually jump and will get copied over onto another gene or next to another gene. And this will create a new gene structure that wasn't found before, right? Which can in turn generate potentially a new function. Okay, so the third one is de novo gene formation, where through random mutation, a part of the genome might acquire a transcription start site, and this transcription start site will then produce a transcript. And then through more random mutation, that transcript can actually gain a start and stop codon, generating an open reading frame, which will in turn create a protein, and this protein can in turn gain potential function and participate in novel mechanism. And then finally, the last one I'm going to present is horizontal gene transfer, right? Which is that in some cases, the DNA from a external organism to you will get close to your own genome, say in a like endosymbiotic relationship or an invasion or an infection, for example. And this piece of DNA will get integrated into your genome. And so with that piece of DNA now present, it'll carry over whatever gene it has on it. And those genes can then in turn be used by the organism that got this piece of DNA integrated into their genome, and then expressed and carry potentially new function as well. And so those are the four more main mode of new gene formation, right? So using this, can we ask a question about like, how did oskar originate, right? And so to answer this, we need to look at the structure of oskar. And so the structure of oskar is made of two conserved protein domain, which is the lotus domain and the osk domain. And those are highly conserved domains, but they're separated by this intradomain sequence, which seems to have no particular conservation and is virtually impossible to align. And so due to that variability, we actually didn't really study its evolution much. Therefore, for the rest of this talk, I'll just only focus on the lotus domain and the osk domain. Okay, so if we look at the sequence identity of oskar, and in particular those two domains, right, we see that the lotus domain has in databases sequences that resembles it, mostly come from other oskar sequences, other arthropod sequences, and other eukaryotic sequences. Okay, and that's not surprising because we know that the lotus domain resembles the Tudor domain, which is a highly conserved eukaryotic domain. Okay. Um, but when we look at the OSK domain, what we see is a completely different profile where most of the closest sequences of the OSK domain are actually coming from bacteria, right? With very few sequences in the center here that seems to come from eukaryotes. So this result sort of, led a hypothesis, which is that could this gene be the product of a partial horizontal gene transfer of this area? And then, like something else happened. And so in order to be able to prove that this area was the result of a horizontal gene transfer, what we needed to do was to use the currently best methodology, which is phylogenetic reconstruction, which infers the evolutionary relations of different sequences, right? And using this, we can try to validate our hypothesis. So I took the sequence alignment, extracted the Lotus and OSK domain from this, created an HMM profile for both of those domains, and then took the Lotus and OSK hits that were found in databases and ran a phylogeny reconstruction with them, okay? So we got a complex phylogeny out of this, and it doesn't fit on the screen, so I'm only going to use a simplified version, which only contains most of the general topology of the phylogeny and is sufficient to explain the mechanism that I'm going to talk about. So for the Lotus domain, most of the Lotus sequences, we find that they are closely related to Tudor 7 from arthropod, right? And this is sort of unsurprising given that we know lotuses, as I said before, are resembling Tudor protein domains. There is the weird case of dipteran lotus sequences, which branch with non-arthropod Tudor 5, and I don't really have an explanation for this at the moment. Okay, but for OSK now, right, what we find is that Oskar OSK actually branches most closely to bacterial sequences. It is nested within bacterial sequences, which is a first good indicator that this is probably a horizontal gene transfer. Moreover, the closest sequences to Oskar OSK in this phylogeny are actually sequences that come from bacterial clades that are known in insects to harbor germ cell symbionts, making this probable HGT even more likely, because if it is in a germ cell, the likelihood of this DNA being passed to the next generation is even higher, right? But what about the alternate hypothesis here, right? Because we could say that, Oskar actually, this topology is wrong and the phylogenetic reconstruction did a mistake, and OSK might actually be more closely related to the other eukaryotic sequences that we find here, and that the bacterial sequences are more closely related to each other, right? There is a test for this, which is called the so what test. It's a statistical test where what we do is we take our tree at the beginning and we put a constraint on it, and we say, okay, the bacterial sequences have to stay together, they have to cluster together, right? And the eukaryotic sequences have to cluster together as well, right? And then we take this constraint and we rerun the phylogeny analysis with those constraints, and we compare the obtained likelihood between both trees, right? And then from this comparison, we can then derive a p-value to know if our topology is highly significant or not. And in our case, we had a really good p-value that claimed that stated that this topology was very likely to be the true topology for OSK coming from a bacterial ancestor. All right, okay. So to summarize, basically in an ancestor of insects, there was probably a germ cell which probably had a germ cell symbiont in it. And in that germ cell, that part of that DNA of the symbiont went and contained the OSK domain and got integrated near a ancestral lotus domain, right? And then through random mutation and random event, this part of the DNA became coding and included this interdomain region that is now this like unconserved region, creating a new gene structure that we now today know as Oskar. All right, so Oskar originated from a horizontal gene transfer, but can we now go back a little bit to germ cells and ask, can we use our data set to try and refine the hypothesis that Oskar became germplasm nucleator of holometabola. Okay, so to do this, we used a fact that transcriptomes contain important metadata, right? And so with those metadata, what we can do is we can extract which tissue and which developmental stage this data set was generated with, right? And from this, we can extract things like the nervous system or the germline, right? Or the egg and adults, and we can then look on the evolution of insects. Where do we find Oskar expressed and at which stage? Okay, so when we look at the insects group, we find that in Ametabola, we find Oskar expressed only in adults, and there was no tissue metadata for that group. In Hemimetabola, we find Oskar to be expressed in the nervous system and in adult insects, right? And only in Holometabola do we find Oskar expressed in the germline as well as in eggs, right? And so this analysis is not complete because there are fewer Ametabola and Hemimetabola sequences. So it might be that in the future, someone might find germline here. However, it is based on hundreds of sequences. And so in comparison to the previous hypothesis that was based on only a few sequences, this result is much stronger to claim that this germline function of Oskar probably emerged in Holometabola. And so to try and understand what are the evolutionary changes that happened to Oskar at that branching event that led to this neofunctionalization, what we need to do is first review what is known in Drosophila about the three main biochemical function of Oskar. Okay, so first, Oskar, through its lotus domain, will dimerize, and the significance of this dimerization is still mostly unknown, but it is hypothesized that it plays a role in the capacity of Oskar to generate germplasm, to nucleate the germplasm. Second, Oskar binds RNA. The OSC domain can bind RNA molecule, and this binding is important for the correct transport and localization of key germ cell determination transcript. It's also hypothesized that this capacity to bind RNA molecule could play a role in the nucleation of the germplasm. Okay, and then finally, the third function I'm going to talk about is that lotus binds VASA, and VASA is a key germ cell determinant that is conserved throughout animals. And by binding VASA, the Oskar protein will then localize this VASA protein at the posterior pole, helping with the specification of germ cells and the germline. All right, so using those three functions, given those three functions, we're going to ask the following three questions. Did Oskar evolve differently between hemimetabola and holometabola? Can we detect a differential conservation of the lotus dimerization? And finally, which amino acids are conserved at the lotus VASA binding interface now that we have so many sequences to study this? And so to study all of those questions and try to answer most of those questions, what we're going to use is a tool called sequence conservation, right? And this is a great tool to study the evolution of protein in general. Because proteins, right, we can align them in sequence alignments, right, where at each position, the amino acid of that particular sequence from an organism will just try to match best the amino acids in other sequences. And using this, we can then take each column, right, and look at whether or not the amino acid at this position changes not at all, like this column, or a lot like this column, right? And so this variation in amino acid at different position allows us to calculate what is called as a conservation score. And for the rest of this talk, the main one that we're using is called the Valdor conservation score, which is a meta score computing things like physical-chemical property, but also statistical property of each position and the evolutionary pressures from the transition between different amino acids. And so using that score, we can then go on each position in the alignment and assign a particular conservation for that position. But we don't have to use a meta score. We can also look at other aspects of of protein evolution, right? For example, amino acids can have electrostatic charges. They can be negatively charged or positively charged or neutral, right? So we can compute the conservation of charge at a particular position, and this is what we call the electrostatic conservation charge. We can also do similar things with the hydrophobic or hydrophilic properties, and we can compute a hydrophily conservation score, which will inform whether or not this particular position was remained in one of those two categories. Okay, so using those, right, using sequence conservation, can we try and find if Oskar evolved differently between hemimetabola on the one hand and holometabola on the other hand? And so to do this, right, we split our data set so that we can now compute the conservation core of holometabolous sequences versus hemimetabolous sequences. And given our assumption that conservation is a proxy for function, right, we can try and compute the bias in conservation. If part of Oskar is more conserved in one of the two groups, this can be a good indicator that it became important for a new function. And so after we computed this conservation bias, right, we looked at the lotus domain and the OSK domain. And what we found was that the lotus domain was centered around 1, which meant that overall, this domain did not seem to harbor any differential conservation between hemimetabola and holometabola. However, when we looked at OSK, right, we saw that the OSK domain was heavily biased towards the hemimetabolous sequences, meaning that OSK is overall more conserved in hemimetabola. So if we look more closely at OSK, so we can see that the bias in conservation on the OSK domain, right, is mostly located on this particular surface of the protein domain. And this is particularly interesting because in Drosophila, this particular patch harbors 3 of the known important amino acids required for the RNA binding property of OSK, right? So does that mean that the RNA binding is actually more conserved in hemimetabola? Whereas our hypothesis was that it actually was probably more conserved in holometabola because of the importance of mRNA binding for the nucleation and localization of transcript. So when we actually looked at the electrostatic conservation score, what we find is a bit of a difference here, because in hemimetabola, right, the conservation of charge, which is necessary for RNA binding, right, is not actually highly conserved at this particular patch of the surface. However, in holometabola, the conservation of a positive charge of the surface is much more conserved, right? And so this sort of leads to a contradictory result here, where it seems that hemimetabolous OSK played probably a different role, at least in this area of the domain, than RNA binding, but that this function required a higher conservation of the surface, right? Whereas in holometabola, right, the electrostatic property of RNA binding are highly conserved, but the overall conservation of the protein domain and this particular patch is less conserved than in hemimetabola. All right, and so with this, we're gonna go on to the next question, which is that we ask whether we could detect a differential conservation of the lotus dimerization. The reason we're asking this is that a previous study has shown that some lotus domain of Oskar do not always dimerize. So in flies or in wasps, right, lotus will dimerize, but in crickets and in ants or mosquitoes, lotus, when put in vitro, right, when expressed in vitro, will not dimerize. And so using this information, right, we then tried to detect a particular evolutionary signal that could be a marker of the dimerization or the absence of dimerization, right? And so to do this, we took our lotus sequences and split them into 2 data sets, like we just did for holometabola, which is the didimeric lotus and monomeric lotus, right? And we computed the conservation in both of those data sets. And we then focused our analysis onto those 2 areas, which are known in Drosophila to be important for this dimerization, which is an electrostatic interaction here between 2 amino acids and a hydrophobic interaction here between the same amino acid on both sides. All right, so when we look at the conservation of those positions, we find that in the lotus that are known to be monomeric, this conservation of this electrostatic property is completely lost, right? We don't find any conservation of charge there. However, when we look at the dimerizing lotus, we find that this particular property is highly, highly conserved. And so if we look at this hydrophobic patch, we find the exact same pattern where the leucine here that was participating to this hydrophobic patch actually reverted to being hydrophilic in monomeric group, whereas in the dimeric group, it is still heavily hydrophobic, right? So to conclude, while the exact function of this dimerization is like still a bit unknown, it appears that the mechanism by which the lotus domain dimerize seems to be at least highly conserved. All right, and finally, the last questions that was interesting was which amino acids are conserved at the lotus vasa binding interface. And so previously, there was different amino acids that were studied at this interface. One of them was this alanine here, and it was shown in Drosophila, right, to be important for the stability of this interaction through a hydrophobic interaction with the phenylalanine that are in front of it. However, our analysis actually shows that the conservation of the hydrophoby is not highly conserved, and even more, it seems that overall, this particular area seems to be kind of hydrophilic in different oskar sequences. But what our analysis also reveals is that we find that 3 other amino acids that are very close to this hydrophobic patch here have a very high conservation of their hydrophobic property. Right. Moreover, some of them are very, very conserved. We also find new potential amino acids, such as this arginine, which might create electrostatic interaction, further refining this particular area, right? And so in this one area of the vasa and lotus binding, right, the conservation of this amino acid is a bit higher. And so here, what you see is a sequence logo. And so sequence logos are a way to represent the conservation of protein sequence in a visual form, where the height of each of the letter represents the conservation of this amino acid at this position, right? The total height of that position represents the total conservation of this position. And the diversity of letters that you can read will basically represent the diversity of amino acids present at that location. And so in green, those are the new amino acids that we're proposing, and in red, those are the other amino acids that were proposed previously. And so what we find is that our new positions are actually have a slightly higher conservation overall than the previously proposed one, and seems to have physicochemical property that makes sense with this interface at this location. Furthermore, when we looked at another previously described important location, which is this alpha helix here, and previously there was this leucine that was described. This leucine is still conserved, right? And so it's probably an important amino acid. But what we find is that there are 2 other amino acids that are way, way more conserved. And by more conserved, I mean that throughout our entire 369 amino acids, 369 oskar ortholog, right, those barely ever got mutated, right? There's like virtually just like a few examples where those positions have ever changed. So they're extremely conserved. And those positions, this histidine is facing a methionine here, which can create a hydrogen bond, right? Which could further help to stabilize this area. And this glutamine here is facing an arginine, which can create an electrostatic interaction, also stabilizing this potential interface. So to conclude, our analysis revealed that new amino acids are probably very important for the binding of lotus and vasa, and those amino acids are more conserved overall than the previously described one, which might reveal a stronger impact of those position for this binding interface. And so in summary, today we learned that oskar is a new gene which originated from a horizontal gene transfer followed by a rearrangement prior to the divergence in zygomenta. The OSK domain is more conserved in the hemimetabolous than in holometabola. Key RNA binding amino acids that have highly conserved electrostatic properties on the OSK domain are actually highly conserved in holometabola. And other key amino acids responsible for the dimerization of lotus and Drosophila are also highly conserved in dimerizing lotus. And finally, we describe new amino acids that are highly conserved at the lotus-vasa interface. And so some future potential direction that this work could go towards is that one, we could generalize the approach to detect the OSK horizontal gene transfer event in other genes, right? So this sort of event within a gene is not something that a lot of tools or sequence analysis methods are looking for. And so it would be interesting to try to generalize the methodology that we applied to a lot of other genes. So that we might give new origination hypothesis for other new genes that are without one for now. Moreover, we could extend this dimerization detection via a machine learning classifier because of this very strong signal that we found in the sequences. We could try to train a machine learning classifier, apply this to all of oskar sequences, and then use this now predicted dimerization or monomeric form to try to further gain an idea of what its actual function in oskar is, given that it's still kind of unknown at the moment. We could also do a coevolution analysis of the lotus-vasa binding interface because this binding interface is probably highly conserved, and therefore it would be very interesting to know if a mutation on the lotus domain actually correlates with co-mutation on the vasa interface, on the vasa protein at this interface. And then finally, could be very interesting to do some mutational studies to try to understand those undescribed amino acids of the lotus-vasa interface, and potentially the one that could be found by this coevolution analysis as well. And with this, I would like to thank all of you for listening to me.